Welcome, everyone. My name is Anna Maria Niewiadomska, and I'm the Global Segment Manager for Public Health and Infectious Disease at Oxford Nanopore Technologies. I'm delighted to welcome you all today to our webinar, where we'll be joined by Gautam Thaku, a scientist on the Rapid Response team at CZ BioHub, and by Georgios Kitsios, Assistant Professor of Medicine at the University of Pittsburgh. Thank you for joining us for today's online session, where we'll be looking at global lessons from metagenomics for future critical care and pathogen surveillance. After the talks are over, the team will be available to take your questions in a live Q&A, so please add them to the tab on the right of your screen. And you can also upvote the questions that you would most like answered, and we'll try to get through as many of them as we can. This webinar is being recorded and will be made available on demand after the broadcast. So you might be asking yourself, why use metagenomics when you can use tried and tested methods for microbiology? Well, when you have a suspected infection after you take your sample, many of these methods can take quite a long time, taking several days and even weeks to get your answer. And they can also be limited in terms of the information they give you. For example, PCR panels are extremely fast, but will only detect a limited number of pathogens that you're looking for, or specific AMR targets, for example. Metagenomics, on the other hand, can give you answers directly from the sample itself without the need for subculturing, and will also allow you to detect taxa from multiple different groups of pathogens, including viruses, fungi, bacteria, and potentially allow you to detect antimicrobial resistance genes as well, providing a much broader coverage than things like PCR and a much faster turnaround time than culture. So many of you may already be familiar with our microbial surveillance metagenomics protocol. This is an end-to-end protocol that we released early last year, which allows you to process a sample by splitting it into two arms, a viral acellular and a bacterial fungal arm, extract your nucleic acids, prepare your library, sequence it on a flow cell, and analyze it through our Epitome software. I won't go into too much details on this because our speakers will tell you more about it. And if you'd like to learn more, you can follow these QR codes to find our protocol, a know-how document, which answers a lot of frequently asked questions, as well as a webinar, which goes into the details of wet lab preparation, as well as the analysis for the dry lab portion using our Epitome workflow metagenomics. And then one more exciting announcement before I turn you over to our speakers. Although you can use our metagenomics workflow in our Epitome software, this uses publicly available databases to find your specific pathogens or find out what's in your sample. However, we are partnering with BugSeq to enable integration of BugSeq workflows within the Epitome software so that you'll be able to analyze your data sets directly within the Epitome application, allowing you to also generate results and a report, a BugSeq report directly within the Epitome application. So you can view your results without leaving Epitome. And this will be released in the next Epitome release within the next few weeks. And if you're interested in learning more about it, please do reach out directly to our BugSeq partners. And I will now hand you over to Gautam Thaku from CZ BioHub to start us off today. Welcome, Gautam, and over to you.
Thank you so much, Anna, for the introduction. Hi everyone. I'll be talking to you about our work using the Oxford Nanopore Metagenomics protocol and our work evaluating it and then also expanding its use by doing trainings across the globe. So this is a quick agenda, so I'll go through a little bit of background and then discuss our results benchmarking the protocol, and then also a little bit of our experience testing clinical samples. And finally, I'll share a few words on our work doing capacity building globally. So starting with some background, So hopefully all of you are here because you are interested in doing metagenomics or already do metagenomics. But just as a quick overview, metagenomics is really a powerful tool for pathogen surveillance, and it's uniquely suited for being able to detect rare and novel pathogens across a range of sample types. And so this makes it really valuable for things like outbreak investigation and so makes it a key part of the toolkit for public health surveillance, but is also used increasingly in clinical diagnostics. So if there are unusual cases where traditional diagnostics aren't able to identify a causative pathogen, then metagenomics is increasingly a tool that clinicians are able to turn to identifying the etiology of disease. Um, and so with that, there's also now really increasing interest in using long read Oxford Nanopore sequencing for metagenomics. And maybe many of you have already used Oxford Nanopore and know what some of its advantages are. Um, and one of them being like a low barrier to entry. So we work with groups across the globe, and many of them are really just starting off doing sequencing, and to them, Oxford Nanopore sequencing is really an entryway, a gateway into the world of sequencing. Um, and so being able to use this platform to do metagenomics has been of growing interest. And of particular interest when we talk about metagenomics for pathogen surveillance is detecting viruses, and in particular RNA viruses, because many emergent uh, uh, outbreaks tend to be caused by RNA viruses. But some of the challenges include, especially when we're working with human samples, the fact that the amount of input is really limited. And so, in the case of human samples, this might be less than 10 nanograms of nucleic acid, and often it's even less than 1 nanogram. So those are fairly low input amounts to use. Um, and RNA specifically is also challenging to work with because of how fragile it is and how susceptible to degradation. Um, and so for this reason, we sort of explored, a few different existing protocols that use Oxford Nanopore metagenomics, and the one that we sort of found most promising uh, was one that uses the rapid PCR barcoding kit. So a PCR step is essential because of the low input amounts we are working with, and and early this year, um, uh, Oxford Nanopore came out with their own variant of this rapid metagenomic sequencing protocol, which is built off of a previously published SMART9N protocol. And so this is the one we've been using now since it's really optimized for the newer R10 chemistry. So this is just, a screenshot of the protocol that you can find on Oxford Nanopore's website, um, and and so I'll just go over it real quick, and I will be focusing mainly on the library preparation part. Um, some of you may be curious about the extractions, and in general, we tend to recommend whatever extraction method you have been using. In our own lab, we use Zymo extraction kits, but have also found other pens to work fairly well. So the main focus here in this slide is the library preparation part, which um, the viral metagenomics protocol makes use of these random um, oligos that bind to RNA, which we are then able to reverse transcribe. And for the reverse transcription, there are these MMLV reverse transcriptases that are used, which have this property of adding additional Cs at the end. And so we can take advantage of this um, property by using what are called template switching oligos, which essentially have complementary Gs at their end. And so um, using this, we can do what's called template switching and end up with complementary DNA that has adapter tags at both ends. And so this is originally a strategy that was used in like single cell RNA sequencing, but in this case has been adapted to viral metagenomics. So in single cell RNA, instead of using random N-mers, you might use um, poly T oligos to capture the poly A tails of mRNA. Um, but here we use random N-mers so that it can capture sort of any RNA sequence. So after this template switching, we then sort of feed into the rapid PCR barcoding kit reagents. So there's a barcoding PCR step, and then pooling and sequencing adapter ligation before loading the sequencer. Um, another thing I wanted to note is for the data analysis. So generally with metagenomic sequencing, there's a lot of data that's generated, and the analysis can be fairly complicated. We use this platform called CZID, which we helped develop in collaboration with Chan Zuckerberg Initiative, and it's a online platform that does not require any coding, and all you have to do is upload your FASTQ files, and the pipeline will run on the cloud, and then you can review the results using a web interface. And there is also a long read pipeline that can be used for Oxford Nanopore data for metagenomic sequencing analysis. Um, this is just going over quickly how the sort of workflow is for the data analysis. Basically, there's a little bit of pre-processing in terms of removing host reads and subsampling if you have like a lot of reads uploaded. And then there's an initial assembly of contigs using Metafly um, and Minimap2. And then those are then aligned to online databases, so the NCBI, NT, and NR databases. So one thing to note is that, yes, like we align to both the nucleotide and protein database. And so this can be uniquely helpful in identifying novel emergent viruses that may be fairly divergent. Um, and then after this alignment, the results are sort of combined and reported out in a simple, accessible way. Um, so, so that's the workflow we used for data analysis. And you can learn more at czid.org. So I'll move on to sharing some of our results now. And first, I'll talk about a little bit of benchmarking that we did. And so in order to do benchmarking, we had a few questions that we wanted to answer. And these are probably questions you have too, like, what is the minimum input required for making good libraries using this protocol? Or what is the minimum abundance of the target that can be detected? And then perhaps also what the accuracy is of assembled contigs. And so to answer this question, we use some synthetic standards. So they're called ERCC standards, and they come in a range of different abundances, and also are RNA transcripts of varying size. Um, and and so using varying amounts of this spike in as input, uh, we observed that we were able to generate what looked like good libraries from as little as 10 picograms of RNA. So on the right, we have a tape station report, which essentially is a size profile. And you can see on the far right that even at 10 picograms input, we're able to see what looks like good library. This is just a QC step, so this is not confirmation. So So after this, we went ahead and pulled these libraries and sequenced on the MinION. Um, and indeed, we were able to get good sequencing data even at the really low input amounts. Um, so, so then the question is, okay, what is the minimum abundance of target that we can detect? And so I first want to introduce this plot, which, as I mentioned, so ERCC is a collection of different transcripts, synthetic transcripts. And so in this plot, each dot represents a particular transcript. And we know going in what the true abundance of each transcript is. And so on the y-axis, or sorry, on the x-axis, what you see is the true abundance measured in bases per million. Um, and on the y-axis is the observed abundance. So this is what we see after sequencing. And as you can see, overall, the correlation is fairly good. There is greater variation at the lower abundances, which is to be expected. But another thing that we notice is that this depends on the total input amount. So, for example, um, if we put in 500 picograms of input, then the correlation is slightly better than if we put in 10 picograms. But nonetheless, even at that low input, we do see fairly good correlation. The other thing is at what point do we start to see drop off? So what you see in red, the red dots, are transcripts that are supposed to be present at low abundance in the input, but that we're not able to observe after sequencing. So they're dropping off. And the point at which the drop off happens depends on the input amount, the starting input amount. Um, but but as you can see, even at the 10 picogram input amount, for about half a million gigabytes of data, all targets that were greater than 100 bases per million in abundance were detected. So this is not to give like a hard um, uh, value or threshold for the minimum abundance that can be detected, but it is hopefully a way to guide how you might think about estimating this for your own application. So then another question is, okay, now we get all of this sequencing data, but what is the accuracy when we assemble them into contigs? And so to do this, again, we took our sequencing reads and assembled them into individual transcripts, transcript length sequences. And then what you're seeing here is the percent identity of those individual transcript sequences versus the actual sequence as a function of the coverage across each transcript. Um, and so what we observed here was that when the coverage was greater than 100 reads, we saw essentially 100% identity. And below that, we started to see drop off, and especially below 10 reads, coverage less than 10 reads, we start to see the percent identity fall off a bit more. But at least again, this gives us sort of something to aim for if we do care about the individual read accuracy, is aim for at least a coverage of 100. Okay, so that was some initial benchmarking we did, and from the outset we were interested in being able to test, actual real world samples. And so we had started doing this early on. And what we found initially was actually that there was a bit of a issue that we encountered, which we're calling here barcode bleeding. But essentially, so what you see here is what we took as the ground truth for a set of nasal swab samples for respiratory surveillance. And so what you see here along the x-axis are different samples, and along the y-axis, different viruses that were present in those samples. So when we ran these samples on Nanopore using the metagenomics protocol, what we found was that it seemed to work well, but we were seeing this sort of crosstalk where, in this example, if you look at the bottom row, there are these spurious signals. So essentially, samples that are not supposed to have this human coronavirus, but seem to appear to have it. And we worked with Nanopore to talk about this issue, and and it turned out that what was happening was that this was um, barcode barcodes that were being read in the middle of the read. So the first thing is that we observed that it occurs um, uh, more frequently when the library size of a particular sample is really short. So generally, less than 1,000 base pairs, maybe even less than 500 in some cases. And also it occurs when one sample in a pool, when we're pooling multiple samples for sequencing, has a really high abundance of a particular target. So if we go back to our heat map, you will see that one of the samples has a really high abundance of human coronavirus. And so that condition results in this um, barcode bleeding, but specifically for that target. And so when we looked at these individual sort of reads, what we found was that this was happening because of uh, like incorrect assignment of reads to their barcodes. And there were reads that had like essentially two barcodes in them, and so the outer barcode was being used for assignment, where in fact, in reality, we would want to throw away a read like this. And um, and so using a filtering step where we remove these mid-read barcodes has really helped resolve this issue. And I hear that the latest version of Minnow is able to do this automatically, but we also wrote a script um, that helps do this filtering prior to data analysis and it's available on GitHub. Um, okay, so with that, we then went on to test more respiratory samples, and this is just a heat map showing how we were able to identify a range of respiratory viruses across these samples. Since then, we've also tested it on serum. So this is, not the largest sample set, but we were able to successfully identify viruses in serum samples as well. Um, so, so a summary of our clinical testing is that, we've found that the Nanopore metagenomics protocol works across a range of sample types. We've tested nasal swabs, serum, and also stool. Um, but that if you are multiplexing libraries or samples in one sequencing run, you do want to be cautious of barcode bleeding. Um, and another thing is that we have noticed that, the N50 read lengths we get from sequencing it tends to be lower generally for clinical samples compared with like synthetic spike-ins or even like culture extracts. So, that's not surprising because clinical samples might be more susceptible to degradation and generally are perhaps lower quality, but it is something that highlights to us that the quality of the sample really makes a big difference. Um, Okay, so in the last few minutes, very quickly, I'll go over, some of our work doing capacity building. So our team, is focused really on conducting trainings globally to help increase sequencing capacity. And so we work with over 20 groups across the world, mostly in the global south. And as part of our training program, over the past year, we've incorporated Nanopore Metagenomics as well. And so we've done, about 4 trainings so far, and we have a few, we have uh, some planned in the future as well. Um, and, this is just some lessons from our work with global capacity building. So, what we've observed is that, supply chain issues for reagents tend to be like a really big challenge. Um, and that having instruments that are really robust to variations in temperature, humidity can really make a big difference. And we're excited about the new, you know, um, MK1D instrument for this reason. And also through our engagements that there's just so many opportunities for collaboration and increasing global capacity. Um, if you want to learn more about our work as well as specific resources like protocols and slides, you can visit our website. There's the QR code here, and I want to acknowledge all of the people who have been involved in the work that I have shared, including in particular my colleagues at the BioHub and our collaborators at Chan Zuckerberg Initiative, as well as Oxford Nanopore, and then of course finally our collaborators across the world. So thank you so much for listening, and I think we'll be open to questions now. And yeah, I hope you have a good day.
Thank you, Gautam, for that fantastic presentation. Gautam will be back later in the broadcast to take part in the Q&A. We now go to Georgios Kitsios from the University of Pittsburgh. Welcome, Georgios, and over to you.
Thank you, Anna, for the introduction and the invitation to present today. My name is George Kitsios. I'm an assistant professor of medicine at the University of Pittsburgh and a pulmonary and critical care medicine physician at UPMC. And my work focuses on understanding the metagenomics of pneumonia and complex clinical syndromes in the ICU. Today, we will review recent work for my group titled Beyond Culture, Metagenomic Insights into Pneumonia Complexity and Future Clinical Decision-Making. These are my research funding disclosures, as well as my role as consultant and member of an advisory board. And these are the disclosures for the institutions that I work for. And these are disclosures from Oxford Nanopore Technologies that the products to be discussed today are for research use only. So as a physician scientist, the clinical questions that I encounter at the bedside are the ones that direct my research in the lab. And a fundamental question when I see patients with suspected infection is, what are they infected with? And in this context, we're trying to develop culture-independent paradigm for pneumonia diagnosis using metagenomics. At the same time, patients with the same syndromic diagnosis can manifest with substantial clinical heterogeneity. And here we're trying to understand what drives this heterogeneity to develop a precision medicine model for acute lung injury. And today we'll examine how these questions overlap and how pathogens may affect the critical illness heterogeneity we see at the bedside. And we'll study pneumonia that was once called as the captain of the men of death. In 2019, pneumonia accounted for 2.5 million deaths globally, representing a deadly, costly condition that drives antibiotic resistance. And since 2019, we've all witnessed the impact of a severe pneumonia caused by COVID-19. But it is really challenging to find the causal pathogen in pneumonia. In this very well done study by the CDC, that they examined systematic fashion in patients with community-acquired pneumonia for the etiologic pathogen. They found that bacterial pathogens were about 14%, viral pathogens about a quarter of cases, but the majority of cases did not have a pathogen isolated despite strong clinical suspicion of an infection. And I submit that at least part of this problem has to do that our current diagnostic framework relies on the centuries-long pillars of microbiologic diagnosis and the fact that we need to culture a microbe and be able to visualize it under the microscope to implicate it as a cause of infection. And this is how this affects the patient care. So when we have patients with severe pneumonia on a ventilator, we are encouraged by clinical practice guidelines to collect respiratory specimens and blood biospecimens and submit them for microbiologic workup with cultures and adjunctive molecular testing. But time is of essence, and we cannot wait for those results that may take 2 to 3 days to come back. So we need to start broad spectrum antibiotics. But even by the time the workup is complete, as we just saw, about 60% of the patients will not have an etiologic pathogen isolated. But these patients are sick, our clinicians are hesitant to discontinue antibiotics, which are often expanded or prolonged, resulting in toxicity, cost, C. diff colitis, and buildup of antimicrobial resistance. And it is in this context that we propose that rapid metagenomics directly in those primary biospecimens can disrupt this problematic diagnostic framework. And we've done some early descriptive work in the field. Initially with Illumina 16S sequencing, we took patients with culture positive or culture negative pneumonia, and we opened that black box of culture negative pneumonia cases and found that about 20% of participants had missed pathogens, high abundance of staphylococcus or pseudomonas, typical respiratory pathogens, whereas the majority of patients had high abundance of typical oral microbiota that would not justify broad spectrum antibiosis. We then tackled a very common clinical problem, the one of aspiration pneumonia, that for a long time has been suspected to be caused by anaerobic bacteria that would not grow in cultures. Well, we used our culture-independent methodologies with Illumina 16S and then some validation with Oxford Nanopore, and found that, in fact, macroaspiration pneumonia did not have any signs of enrichment of those implicated obligate anaerobes. And then in severe pneumonia cases with Oxford Nanopore, we applied a host DNA depletion method to be able to show some proof of feasibility data that this can be done in patients in rapid turnaround times. In 2025, the state of the literature shows that there are only a few studies published around the globe with respiratory metagenomics for pneumonia diagnosis, and most of them clusters in different centers in China. And I think that the lack of adoption of metagenomics does not only have to do with technical difficulties. I think a fundamental challenge has to do with the diagnostic framework. And the fact that our reference standard is not perfect. And we consider that culture-positive cases are gold standards, but then on those culture-negative cases, we really do not have any way of confirming that unknown diagnosis. And expanding a little bit more on this, we have this framework that we consider culture-positive cases as a reference standard. And we expect then, based on what we know from the ecology of pneumonia and the respiratory microbiome, that is shaped by increased immigration of bacteria from the upper to the lower respiratory tract, decreased elimination, altered environmental growth conditions, that in the context of pneumonia, we would have high pathogen abundance displacing that low abundant, highly diverse, low biomass microbiome of health. And to prove this framework, we're now going to study the case of Staph aureus pneumonia as the gold standard of respiratory infection, and Staph aureus ironically got its name by the Roman gold coin of aureus. So this is going to be a reference standard and see how this paradigm shapes the metagenomic profiles we expect. And Staph aureus pneumonia is an important clinical problem. Even for community-acquired pneumonia accounts for a substantial proportion of culture-positive cases, but it is in the hospital, in hospital-acquired pneumonia and ventilator-associated pneumonia, that it is a major pathogen that is highly virulent, causing hemorrhagic and necrotizing infections associated with poor outcomes, and mortality that at times exceeds 50%, and also challenging to treat organism as there is widespread resistance. And that's why we conducted this study at the University of Pittsburgh. So we selected 107 adult participants that had enrolled in a prospective registry in the ICU. Those participants were all invasively and mechanically ventilated through an endotracheal tube and had respiratory specimens collected for clinical purposes with suspicion of pneumonia, and we included only participants that had Staph aureus isolated from those respiratory cultures. So the gold standard diagnosis of Staph aureus pneumonia. All those cultures were collected within 72 hours of intubation. So from those participants, for a random subset of them, we also collected research biospecimens from the respiratory tract, non-invasive endotracheal aspirates, from which we extracted genomic DNA in the academic research lab. And from that DNA, we performed either Illumina MiSeq 16S sequencing and also Oxford Nanopore metagenomics with a MinION device and analyzed data with a BugSeq platform. Those participants had a median age of 55 years, a high proportion of comorbid conditions, and really a sick cohort, 29.4% 60-day mortality. First level of analysis was looking at those clinical microbiology cultures. And what did it tell us? First of all, we classified the growth level of Staph aureus on those petri dishes in semi-quantitative fashions as reported by the clinical laboratory into rare growth, light, moderate, or heavy growth. And also looked whether Staph aureus grew alone on the petri dish or whether there was evidence of other co-growing organisms, either normal flora, yeast, or gram-positive or negative pathogens. Importantly, we found no significant differences in clinical characteristics between these subgroups defined by culture results. And also, these culture results did not predict survival. We found no significant differences in 60-day survival by Kaplan-Meier curve analysis between growth levels of Staph aureus on the petri dishes. Whereas by growth pattern, perhaps there was a trend for some delayed worse survival in those cases that only grew Staph aureus. Nonetheless, it was not statistically significant. Which prompted us to go then to the next level and now go into Illumina 16S. Now we take the 16S profiles of those 54 cases and we plot the relative abundance of Staphylococcus genera. The heterogeneity is striking. We see that some cases have complete dominance, 100% abundance of Staphylococcus, or others it's barely detectable, below 0.1%. And in fact, we discussed that we were expecting to see dominance by Staphylococcus in those culture-positive gold standard cases. And we found that to be the case in only a third of them. Only 33% of the cases had more than 50% relative abundance of Staphylococcus. So let's examine this a little bit more. We compared 16S profiles with clinical cultures, first in terms of growth level. And we found that there was no difference in relative abundance of Staphylococcus by growth levels of Staph aureus in corresponding clinical cultures, or any difference in the raw number of 16S reads assigned to Staphylococcus genera. However, in terms of the growth patterns, we found that cases that grew Staph aureus only had higher relative abundance of Staphylococcus in corresponding microbial communities. We're not able to find any specific pattern in terms of other co-growing organisms in the relative abundance of Staphylococcus. But this clearly told us that when we see Staph aureus only in a culture, it's very likely that this is dominating the corresponding microbial community. Very important finding that we had 7 cases of Staph aureus bacteremia aligned with those pneumonias. So those cases had higher relative abundance of Staphylococcus in the corresponding microbial communities in the lower respiratory tract. So sicker patients with bacteremia and sepsis have higher levels of Staph aureus in the respiratory communities. We also looked at those research biospecimens for host response biomarkers of this infection. We found that cases with Staph aureus dominance, more than 50% relative abundance, had remarkably higher levels of pentraxin-3, a CRP-like molecule that signifies a response to bacterial infections. And finally, we found that patients that had dominance of Staphylococcus had markedly worse 60-day survival compared to those patients that had Staph aureus pneumonia clinically, but less than 50% relative abundance. However, we recognize that 16S is a proof of concept technology that has some PCR amplification biases, limited resolution, only for bacteria, cannot be done in real time. That's why we expanded with nanopore sequencing and metagenomics with MinION devices. In some earlier work, we demonstrated how we can handle the big challenge of signal-to-noise ratio in human respiratory metagenomics, where the sequencing output can be completely overwhelmed by human DNA. And with a saponin-based depletion assay, we're able to amplify the amount of microbial DNA available for sequencing. We used a rapid workflow, the rapid PCR backcalling kit. Following depletion and DNA extraction, we performed tagmentation and PCR and built pulled libraries between 6 to 12 barcoded samples per run. We sequenced them for about 24 hours on benign devices, and then took the FASTQ files and processed them through the BugSeq platform. And this is what we found by Nanopore metagenomics now. Now we can get down to species level. We got Staph aureus reads, and we found a similar pattern to 16S of tremendous heterogeneity and variability in Staph aureus abundance. We found that about a third of those cases had high abundance of Staph aureus in terms of absolute numbers of reads. And there was a nonlinear relationship between absolute abundance and relative abundance of Staph aureus, taking off at about 500 absolute reads. In terms of relationship to cultures, we found no difference between growth levels or growth patterns with the absolute number of Staph aureus reads. However, when it came to relative abundance of Staph aureus, we found again that cases that grew only Staph aureus in culture had much higher relative abundance of Staph aureus reads by Nanopore metagenomics. And in terms of survival, we found that those cases with high absolute abundance of Staph aureus by Nanopore had significantly worse than 90-day survival compared to the 66% of the cases that did not. We built a Cox model to adjust for age and sex and found that high Staph aureus abundance was the only independent predictor of worse survival at 90 days. And to illustrate this further, we'll review two clinical cases with their course and Nanopore profiles. So case one is a 47-year-old man with diabetes, GERD, who was admitted with COVID-19 pneumonia and ARDS, requiring intubation, neuromuscular blockade, and proning. Very sick patient. He received dexamethasone and IL-6 blockade as it was appropriate for COVID-19 pneumonia. Case two was a very similar presentation, a 45-year-old man with obesity, similar clinical findings, similar treatment. And we can see also in terms of radiographic densities by those RAIL scores, they were also quite similar at the beginning. So let's see what happened. Case one was intubated, was hypoxemic, requiring proning, not getting better, so the clinical team started getting concerned about a superinfection. On day 3, they performed a bronchoscopy that demonstrated heavy Staph aureus growth. I was resistant to clindamycin, sensitive nonetheless to oxacillin. They started appropriate antibiotics, and the patient improved to the point that they were discharged from the ICU by day 11. Now let's see what happened in a respiratory biospecimen that we did Nanopore metagenomics at the same time. Processing this through BugSeq, we found high abundance of Staph aureus and also some oral taxa, as well as high levels of detection of the ERM-A gene that confers resistance to clindamycin. So pretty good agreement with the cultures. This patient had an appropriate diagnosis by both methods and improved with antibiotics. If we look at the output by BugSeq, reports high levels of human DNA, high abundance of Staph aureus that is flagged by an AI summary as the most likely pathogen. We also see that there is a reporting of the antibiotic resistance genes, and we found that this was predicted to be clindamycin as confirmed by the clinical antibiogram. We did not find the MEK-A gene that would confer MRSA resistance. We did not find that. And also there was some reporting of potential virulence factors. But let's see case 2. Case 2 was evolving to be more sick. The patient was severely hypoxemic and required ECMO support by day 2. An early bronchoscopy grew Staph aureus and Klebsiella pneumonia. The patient continued to deteriorate, developed severe necrotizing pneumonia, as we can see also here with many lucences in the lung parenchyma. A second bronchoscopy at the time showed only some rare gram-negative rods and gram-positive cocci, yeast, and normal flora. So the impression that whatever was there was being killed was not really causing more harm. Nonetheless, the patient succumbs to multi-organ dysfunction and severe necrotizing pneumonia with respiratory failure. And by day 12, an autopsy is performed that grows Candida albicans. Let's see what parallel metagenomics told us. We did this all in retrospect, but at the same time, on day 1 sample, we found high abundance of Staph aureus that was also predicted to be resistant to clindamycin, consistent with cultures. By day 5, the Staph aureus abundance is further enriched, also posing this participant in that high Staph aureus abundance tertile with adverse prognostication. But we also see emergence of abundance of Candida albicans, consistent with yeast that was seen on the clinical cultures. By day 10, we have complete dominance, astronomically high numbers of reads belonging to Candida albicans, which we know was eventually recovered from this patient's autopsy in the context of a severe necrotizing pneumonia. So we have two opposing stories, a case of a survivor where we diagnosed Staph aureus MSSA pneumonia, and the participant did well. And another unfortunate case where Staph aureus pneumonia was also found by metagenomics, but eventually a high abundance of Candida albicans progressively detected and recovered on autopsy. On the clinical surface, both cases had severe COVID-19 pneumonia with Staph aureus superinfection. On the metagenomic analysis, we see those stark differences in evolution. So in conclusion, we see that metagenomics reveal hidden complexity even in culture-positive Staph aureus pneumonia, which are actually cases of presumed diagnostic certainty that we take as gold standards. However, pathogen abundance and the surrounding microbial community correlate with host inflammation and survival, now uncovering prognostic heterogeneity that was masked by the clinical culture results. We do need prospective studies to determine whether metagenomic insights can improve antibiotics decisions and outcomes. And we believe that rapid metagenomic integration into ICU workflows represents the next frontier in precision management of severe pneumonia. At this point, I would like to thank the big village of colleagues and collaborators that helped conduct this study and analysis, specifically Dr. Benos and Mr. C, who helped with the analysis for this project, the funders and supporters of this work, but above everyone else, the patients and the families who participate in our observational research completely with altruistic perspective, try to help others in the future during their most difficult times of their lives. And with that, thank you for your attention and happy to take any questions.